Hello and welcome to the walkthrough of the Year 10 mind map for Semester 1 2026. First of all, there's DNA. DNA is composed of nucleotides. A nucleotide is composed of a phosphate, a deoxyribose sugar, and one of four nitrogenous bases, being adenine, thymine, guanine, and cytosine. DNA is double-stranded. Each side is complementary, which means it's matching. It uses the base pair rulings of A goes with T and C goes with G. There's hydrogen bonds between these two base pairs. DNA is also anti-parallel, which means the two sides are parallel but running in opposite directions. Next is DNA replication. As we said, DNA is double-stranded. It has the template strand that runs 3' to 5', and this holds the genetic code. The other strand is called the coding strand, and it runs 5' to 3', and this is copied into the lagging strand. The two enzymes we need to know are DNA helicase that breaks the hydrogen bonds to separate the template and the coding strand, and DNA polymerase that aligns the complementary nucleotides according to Chargaff's rule, which is A goes with T and C goes with G. Next, we have mitosis, which occurs in somatic cells. It can be remembered as IPMAT. It involves one division. It creates two identical diploid daughter cells, and there is no variation between the generations or between the two daughter cells. It's perfect for a stable environment. The steps are: in interphase, the DNA is replicated from single-stranded to double-stranded. In prophase, the nuclear membrane breaks down, and the centrioles start to form the spindle fiber. In metaphase, the chromosomes line up at the equator with one double-stranded chromosome on each spindle. Then there's anaphase, and the chromatids move to opposite poles. And then there's telophase, in which a nuclear membrane forms around each pole, and cytokinesis occurs, which splits the cell into two daughter cells. Mitosis is used by somatic cells for growth and repair. Next is meiosis, and this occurs in the gonad germline. The gonads for males are the testes, and the gonads for females are the ovaries. This can be remembered as IPMAT-PAMAT. There are two divisions. 4 non-identical daughter cells are created. There is a huge amount of variation between the generations. This is perfect for a changing environment. The steps proceed as this. In interphase, the DNA is copied from a single-stranded into double-stranded. In prophase 1, the nuclear membrane dissolves and crossing over occurs. And we'll go through this later. In metaphase 1, the homologous chromosomes line up at the equator and independent assortment occurs. In anaphase 1, the recombined chromosomes move to opposite poles, and in telophase 1, the nuclear membrane forms at each pole. Then cytokinesis occurs, where the parental cell splits into two daughter cells. But now we have to go through the second division. In prophase 2, the recombined chromosomes become shorter and fatter. In metaphase 2, the recombined chromosomes line up the equator, and independent assortment occurs. In anaphase 2, the recombined chromatids move to opposite poles. Then we've got telophase 2 and cytokinesis, producing 4 haploid daughter cells that are all different to each other and different from the parent cell. Now we come to the terminology for genetic crosses. A gene holds the genetic code for a trait. An allele is a variation of a gene, like big A and little a. Both of them are called alleles. The genotype is the alleles possessed, like big A, or big A, little a. The phenotype is how the genotype looks. Homozygous means the same alleles, like big A, or little a, little a. Heterozygous means different alleles, like big A and little a. Dominant means it's always expressed. Recessive means it's only expressed when in the homozygous recessive. Autosomal means it occurs in the autosomes, which are the normal chromosomes, sets 1 to 22. Sex linkage means it occurs in the X and Y chromosomes. Hemizygous means different sex chromosomes, like X and Y. F1 is the first generation of offspring. F2 is the second generation of offspring. Complete dominance is in the heterozygote, which is big A, little a. It shows the dominant phenotype. In incomplete dominance, in the heterozygote, the individual is a blend of the alleles. Like when we cross a red flower with a white flower, we get pink flowers. And codominance is when, in the heterozygote, both of the alleles are expressed, and this is like a roan cow that has red and white splotches. Now we come to genetic crosses. We use a Punnett square, and this calculates the potential outcomes, not the actual outcomes. This provides a genotypic ratio, which is a ratio of different genotypes, like big A to big A to little a. And it also provides the phenotypic ratio, which is the ratio between the outcomes or how the individuals might look. When we do a cross on a Punnett square, the maternal alleles go on one side and the paternal alleles go on the other side. One allele from each parent must meet up, and we do this through the Punnett square. And this shows us the results of a heterozygous cross, which is big T little t crossed with big T little t. And we can see the genotypic ratio is going to be 1 to 2 to 1. 1 big T to 2 big T little t to 1 little t. And the phenotypic ratio, because the dominant wins out, is going to be 3 dominant to 1 recessive. Next, we have pedigrees. A pedigree is a diagrammatic representation of a family tree. The generations are in Roman numerals. The individuals are numbered from the left as in 1, 2, 3, and 4. A square is a male, a circle is a female. Shaded means it's affected, unshaded means it's unaffected. The rules for solving the pedigrees. When it's autosomal, there's roughly equal amounts of males and females affected by the trait. And this is carried on autosomes. In sex linkage, there is sexual bias shown in the pedigree. If there's more males, it's likely to be X-linked recessive. If there's more females, it's likely to be X-linked dominant. When it's dominant, it does not skip a generation. Every affected child has at least one affected parent. And if the trait is recessive, it does skip a generation. 2 unaffected parents can have an affected child, where both parents are heterozygous, being big A, little a. 25% of their children should have little a, and express the trait. Then we went through natural selection. There are 3 people of note. Jean-Baptiste de Lamarck, he suggested that a species changes over time and that frequent use of a structure makes it grow. Traits are passed on to the offspring, but traits are not genetic in origin, but more contained within the organism. His example was the giraffe who wants to reach leaves up the top of a tree, so he makes his own neck grow. Next is Alfred Wallace. He had very similar ideas to Charles Darwin. He believed in variation in a population, that more offspring were born than can survive, and had the basic idea of selective advantage, and he wrote a letter to Darwin with his ideas. Charles Darwin, though, came from a very famous rich family. He had a very famous grandfather called Erasmus Darwin. He invented the tenets of natural selection, of overpopulation, where more offspring are born than can survive, heritable variation, where mutations are passed on from parents to offspring, competition, which is the fight for survival, and survival of the fittest, which is if you have the correct mutation, you obtain a selective advantage and that will always win. And if you're alive, you can breed. He also wrote Origin of Species. His famous work was The Finches at Galapagos Islands. Important in your course is the sources of variation. A mutation is a permanent change in the genetic code, which is the sequence of nitrogenous bases on the template strand. A point mutation is a change in one nitrogenous base. Point mutations occur during DNA replication because the DNA polymerase makes errors and they aren't fixed. There are several types of point mutations. Substitution, which changes one base, say from an A to a T. Addition adds an extra base. Deletion removes one base. The effects of these point mutations can be silent, which means there is no change to the protein made. Missense changes the protein that is made, and nonsense means the protein is made too short. Then we've got crossing over. Crossing over occurs in prophase 1 of meiosis. It occurs between homologous chromosomes. Homologous chromosomes have the same genes in the same place. The non-sister chromatids of the homologous chromosomes overlap. The place where they overlap is called the chiasma. They swap the alleles between the homologous pair. This results in the reshuffling of genetic material from the mother and the father. Therefore, each gamete will have a unique combination of alleles. In independent assortment, this is where the chromosomes line up randomly at the equator, and because there's no set rule, they do it randomly. So there are many combinations possible when they do pull apart and move to each pole. Again, this results in a reshuffling of the genetic material, adding variation to the population. Next is the idea of selection pressures. A selection pressure is a question posed by the environment. So there's variation in the original population, and organisms compete to survive. Those with the correct adaptation, being in the correct mutation, will solve the selection pressure and live. This is called the selective advantage. Selection pressures can only see phenotype. They cannot see the genotype or the alleles you possess. The phenotype is how the genotype is expressed. It's how the individual looks to the environment. If an organism is able to solve the selection pressure, it will live and breed and pass on its DNA to the next generation. And if it can't solve the selection pressure, it will die, decompose, not passing on its alleles to the next generation. And the famous example here is the peppered moth in the Industrial Revolution. An adaptation is anything that helps an organism to survive. They are caused by mutations. They are used to solve selection pressures. And once again, the adaptations can be structural, physiological, or behavioral. And these will be passed on to the next generation. In antibiotic resistance, the original population bacteria had lots of variation due to mistakes in DNA replication. The bacterial DNA mutates rapidly. So there's heaps of variation, and some of that variation will allow the bacteria to solve the selection pressure, because the selection pressure here is the presence of antibiotics. If you do have the correct mutation, you will live and you will pass on your genes to the next generation. If you don't have the correct mutation, which means you don't have the antibiotic resistance gene, you will die and you won't pass on your alleles to the next generation. So the entire population now is resistant, and that's not good. Now we come to the idea of speciation. In the original population, there is variation. Variation is caused by mutations, crossing over, and independent assortment. There are more offspring in this original population than can survive, and there is gene flow within the original population. Then the population splits in two. The two subpopulations are separated by a geographical barrier, like a mountain range. Each population can't access the other, and we call this geographic isolation. Now, two distinct gene pools exist, each with different environments. The different environments means that there is different selection pressures. And if you've got a different selection pressure, different mutations will be selective advantages. The different mutations build up on each side of the geographical barrier in the separate gene pools. Over millions of years, the two gene pools become very, very different with many different mutations. So if they were to come back together after millions of years, they would not be able to interbreed, and we call this reproductive isolation. When one species has become two, we say that speciation has occurred, and here I've misspelt occurred quite badly. Remember, the definition of a species is a group of similar individuals that are reproductively isolated that can produce fertile and viable offspring. Fertile means you can make gametes, and viable means you live long enough to use them. The last thing on our list is genetic diversity. Populations need genetic diversity to solve the changing selection pressures. Without genetic diversity, any small change in the selection pressures will wipe out an entire population and make them go extinct. Outbreeding and random mating are good for genetic diversity as it increases the genetic diversity, and it's great for changing environments as at least some of the members of a species will survive the change and go on to reproduce and repopulate the species. Inbreeding and artificial selection are bad for genetic diversity. But if the population is already suited to a stable environment, it can also be a good thing, but generally it's bad. Okay, thank you for paying attention. Goodbye.